So I will share my. Well, it's not the best PowerPoint. Click here to register.
Mm-hmm.
I think I'll start here.
Mm.
Oh, and it's not. Tony, you are. I'll say you are coming instead of her. We didn't get a new one, so I'm sorry, Tony.
Oh, that's fine. I think we're live. Is this on now?
Yes.
Here we go. You can start. Sven?
Yes. Yeah, hello, my name is Sven Sommer. I'm from Aarhus University, and I'm the co-chair of this manure management network. And today we have organized this webinar called entitled B0 for IPC inventories and biogas production. And it's a very important issue, this B0, because it's a variable that is used both for calculating methane emissions in inventories throughout the world, and it's also used by biogas plants to assess what is the value of the biomasses they are using or buying for their biogas production. And very briefly about myself, I am from Aarhus University, a professor. I've been working with manure management for almost half decade, and I'm now the co-chair of one of the co-chairs of the manure management network after Tony van der Werden, who had done a marvelous job the last many years. So today we are having three presenters, and the first of them is, if he's here, Sasha Hafner, or is he there? No. So the first presenters will be, and will not be April Lightham, but Tony has agreed on giving a presentations on behalf of April, who couldn't be here today. And Tony, he is a researcher, senior researcher from New Zealand, from ag research in the south of New Zealand, and he has been working on N2O emissions and ammonia emissions from land, from fields for 30 years, and he will now give a presentation about his recent project he's carried out, being on the potential for improving a project he has worked on with B0 and about how to which can be used to improve calculation. So Tony, it's I don't know how to. It's not that you're in now to give a presentation.
Nice one. Thanks, Sven. I'll just set up my slides. I probably just need to close down your show, then I can open up mine.
Yeah, sorry. Did I turn it off?
Brilliant. Yes, you have. That's cool. Okay. Yeah, sorry, people, we'll get there. We're just getting a few things sorted here. So that one there. Yes, there we go. We've made it. So yeah, hi everybody. I'm speaking here on behalf of several of us, and April Latham from USDA. She couldn't make it, so I'm speaking, sharing this talk on her behalf. So this is a piece of work that we're proposing to look at. My colleagues are Marianne Holt-Cantillo from Costa Rica, and then Marta Alfaro, she's with me in Ag Research from Chile originally, and then April in the States, USDA. So in terms of what we're talking about here, when it comes to manure management and the impact that it has in terms of global agricultural emissions, we are around that 7% mark, and it represents about 4% of global methane emissions. To account for this for inventory purposes, there is a tier 1 approach for countries that see manure management as a minor source, but we're focusing here on countries that see manure management as a key source, so tier 2 methodologies. That uses, to calculate these emissions for tier 2, We're using information on the volatile solids content, which is the VS there, as well as, and that can be predicted from dietary information such as energy and digestibility. So that's already, there's a predictive model for that one. Then we have the maximum methane potential of manure, B0, and that's the focus of this particular seminar. And the third one is the methane conversion factor, which is sort of like a choke. So the maximum methane potential is really that potential, how much methane can be produced from this particular manure source. Then the the methane conversion factor is a correction for that. It's a percentage reduction according to whether it's being solid separation, or if it's being stored in an anaerobic lagoon, or whether it's a pasture based grazing system, for example. Now, when it comes to the B0 maximum potential for methane, the IPCC currently has a single value for dairy systems of 0.24 cubic meters of methane per kilogram of volatile solids. That's just a single figure that is used across the globe. There is a value for high production systems, which is still 0.24, which you can see on the right there, and then for low productivity systems, 0.13. So the thing is, we sort of see that as being a little bit out of date, and there's potential for improving on that markedly. It's based on old data and very limited data, so it's not very representative of the farming systems that we might have around the world today. April has done some work in the US, so you can see some data on the left here, some preliminary data from dairy manure systems in the western US, and here on the figure you can see the 0.24 is the red dotted line, and she's seen a difference for fresh dung from different age classes of dairy cattle. So dry cows and heifers, quite a lot lower at 0.16, and that's significantly different from the lactating cows, which is close to 0.24 at 0.22. The error bars here are the standard deviation. So if you can imagine, the standard errors would be approximately half of that. Then if we were looking at fresh slurry, the value is quite a bit higher, and that could be a result of the fact that slurry is a mix of dung and urine. So maybe the nitrogen in the urine is having some sort of impact on the microbial activity that's generating methane from the dung. So the value of 0.24, it's a little bit antiquated, and so we see the need for updating those figures for not just dairy or cattle, but also we need to consider different all sort of livestock, main livestock classes. The other work that April has been looking at is developing potential relationships, empirical relationships between the dietary characteristics and B0. So here we see a couple of examples of unpublished data that she's generated, where we've got dietary characteristics such as the acid detergent fiber and neutral detergent fiber and lignin and starch being used to predict B0 values for those dairy manure systems. Now, these types of inputs are already being used for estimating enteric methane, but here we've got an opportunity to go to another source of the methane from livestock systems, how much comes from the manure. If we can do this, if we can establish some solid relationships here, then we can estimate B0 directly from the characteristics of the diet, and that provides opportunities to be able to identify mitigation options, mitigation opportunities, for example, as well as being able to provide a better, more accurate calculation for national inventories. So what we wanted to share with you here was a potential project. We see the opportunity really sits with updating that B0 value, and that would help to improve national inventories. And by including feed analysis, then we've got the opportunity to predict B0 and also help to identify mitigation options. The third aspect of this project is really around being able to support countries in where these improvements are needed to improve their inventories. The proposed project is really focusing on, well, there's several steps that we're sort of looking at. We want to identify countries requiring resources to better quantify B0. We're looking at, and we're probably going to focus on global south countries for this, because we feel that global north countries have got the opportunity or potential to self-fund or partially fund inclusion within this type of project. Second step is determining which countries can utilize centralized laboratories. So we've had discussions with the GRA in terms of the climate smart agriculture measurement hubs, utilizing these hubs that have been set up recently. And there are also other laboratories set up, obviously, around various countries that could be set up or have the equipment and the expertise to conduct these tests in terms of running the protocols that we want to develop for establishing B0 values. The development of the protocol that will work with, we want to develop a method and gather the equipment, make sure that it's all leak proof, and then send that out to the countries that are participating in this project. So we've got a bit of a plan. We're sort of trying to tie off many loose ends around that. The fourth step is providing resources and potentially training, and that could be through a training video, to help the laboratory staff to conduct the methodology correctly so that we have consistency and repeatability across the different labs that are involved. The fifth point here is identifying the key class species per country. So looking at these world maps on the right, you can see the cattle density is quite different from swine density. And at the moment, we are thinking of including both cattle and swine. We just have to see what sort of cost is associated with trying to go with those two very large livestock classes. We may need to just come back and restrict it to cattle. We'll see how we go on that one. And then the next step there is classifying the results. So once we've got data coming in, how can we group it? Do we group it according to diet, or do we group it according to species or production system? We don't know yet. It depends what the data looks like once it comes in. I should also point out, once we've got this project up and running, if we get the funding approved, we would see countries going out into the field, going into the local farming environment, and collecting feed samples as well as manure samples. So we are analyzing feed samples for various characteristics, and we want a range of different diets there to get a good spread, of course, and then the associated manures that are a result of consuming those different feeds. Last step, of course, analyzing that data to update B0 values for country-specific emission factors and inventories, and developing those predictive models. Where have we got to at this point? We have developed a survey which has been sent out to various researchers around the world, and we've collated those results, and that has helped to determine the direction of this project. That's been a critical piece of information, and we see more work to do there. We need to go back out to specific individuals and institutes to gather more information, because what we might find is some laboratories are well established and don't require the equipment that we might have to assemble for the protocols to be carried out, whereas others may need a lot more assistance. So it's going to be very much country to country variation there. Identifying scientists across the globe to conduct this work, that's we've been doing that already, of course. We've got quite a bit of interest from various countries, and through that determining the existing capability and resources for the project. Establishing partnerships with experts such as Sasha and Henrik, the other two speakers in this webinar, I feel is important, that we bring in people that have worked in this space to provide guidance, to add value to what we're proposing to do here. And then the next point is really identifying those potential protocols. Now, we might be utilizing the methodology that Sasha will share in his talk. He's been very helpful with us so far. We've had a couple of chats with Sasha in terms of pitfalls with different protocols. And so we need to be aware of those before we decide on which protocol we decide to go with and send out to these different regions. We are in discussion with the Global Methane Hub. So this is ongoing and we're hoping that something will be successful there, but clearly we are still continuing those discussions. And as I mentioned earlier, we're currently suggesting that the funding will be going towards key global south countries that are representative of major livestock regions of the world. So we're talking Africa, we're talking Asia, we're talking Latin America. So my final slide is watch this space. We hope to start this work, the actual work, in later this calendar year. If you wish to stay informed, or you may even have data that could be useful for the data set that we want to assemble here, please get in touch. Now, for countries that are probably, I guess, more in the global north region, you may be able to source funding to be able to be part of this project. And so please reach out to Marianne Holcantello. She's the lead researcher on this. So yeah, please note her email address there. And if you've got any questions later on in this webinar, then I will attempt to answer them, and I may need to record them and get in touch with you later with suitable responses from one of my colleagues. Thanks very much.
Thank you very much, Tony. The audience cannot see me because my camera is not functioning, and so I'm the black ghost here. But now there are time for some questions to Tony, and maybe I could start. Yeah, well, I'm your chair, so I will just ask you. Do you have some intercalibrations? Because everybody knows there will be so much difference between how people are managing the experimental work, and that might affect your measurements. So intercalibration might be very useful. Is that something that's included in your work?
Yeah, that it is. It will depend on the method or the protocol that we end up decided to go with. So if it was a more of a traditional sort of batch chamber approach where we require inoculum, we'd want to try to ensure that the inoculum was sent, the same inoculum is sent to each lab, so that we can get a trying to reduce the number of variables, so that when we do those comparisons with the labs, that we've got some robustness to that comparison. Now, we will, once we've heard from Sasha, we might find that that type of interlab comparison it's still important. So what we wanted to do is, again, like I said, depending on the methodology, but we want to be able to, at this stage, what we're proposing is bringing in the kit, assembling it all here in New Zealand, testing it for leaks, making sure that it's all leak proof, potentially developing a training video. The other option is to actually go on site where required, to travel to specific labs that may require additional assistance. There's a cost obviously associated with that, but these are all things that can be discussed. But yeah, that interlab comparison is important.
Yes, thank you very much. I don't know if there are questions from the audience. It may not. It's because I have one more question, Tony. Oh, there's one here, I think. No, from Sasha.
No. Oh,
He was raising a hand.
I'm not too sure who
Okay,
raised the hand. I'm still searching,
but then another question is measuring these other this April later, what she was doing was carrying out this Van Soest characterization of organic matter. Are you sure? Is it very precise? Do you think it's more precise to do that than to measuring B0? And
I'm not quite following, Sven. What do you mean?
April Lighton, when she was developing a characterization of the, let's say, manure, it was a fiber fraction she was measuring, the lignin, the neutral detergent fibers, etc., etc. And then she developed a model to calculate B0, maybe including animal category. Are you sure? Do you think that by the end of the day, this will be more precise than measuring B0?
Well, we'll be measuring B0, and the opportunity is to see if we can predict B0. So it's we don't know if it wouldn't be unlikely to be more precise, but it provides an opportunity to be able to predict without having to go through the process of measuring B0 eventually. Yeah, so it's a
Yes.
two-step process.
Yeah. Good. I think that's my questions. And I would just say I'm really glad to see that you have this focus on the global south, because when we look into also the dataman studies, it's obvious that there's a need for more measurements in the global south. And there's also
Absolutely.
competences in the region who can do it. So that's great.
Absolutely right, Sven. There's a massive gap there. That's one of our biggest gaps, gathering data from global south countries for from greenhouse gas emissions from manure
Yes.
management systems. So this would present an opportunity to help to improve that balance.
Yeah.
There's a question in the Q&A. It goes, will you develop a separate diet relationships for pigs and ruminants?
Yeah, well, look, we will investigate that once we've got the data set. It's possible that it's more based on diet rather than livestock type, but I doubt it. I think we would see different relationships for those two different livestock types, but we'll definitely explore the opportunity to combine all of the data into one large data set, and maybe it's a more powerful predictive model. I think we would see them being separate, but let's have a look once we've got the data.
Great. Harry, I hope your question has been answered perfectly. Can I give Harry the platform to talk, because he raised a hand? Okay, let's talk. Harry, you can unmute and talk.
No, the answer was fine. Sorry, I pressed the wrong button. I wanted to press thumbs up. But Tony, the reason why I asked the question is you've got different diets in pigs. You've got a monogastric versus a ruminant, and I presume it can give rise to quite different constituents within the manure. And my question really was, I would doubt if you had a single equation. I suspect you may have to think of a different equation. And then I also think of how, particularly in intensive pig systems, it's a very manufactured diet that is very common. Whereas if you go to the global south, it might be much more varied. And so I was just wondering if it's going to be quite complex to develop diet relationships when you get such varied diets and you have varied species, particularly if it's monogastrics, ruminants. And so I could see somewhere like the US where you've got quite manufactured diets, when you're going to have 30% minimum fiber, you're going to have this. But if you go to the global south, you'll see a much greater range of dietary components, and the whole diet is of a completely different level.
Absolutely. Yep, you're quite right. Yep, but I guess it's and that's why it's important for when we identify these countries and institutes that we get a range of diets, that they go out and collect a range of diets and the associated manures. Otherwise, we need that spread to see if there's if one can be a predictive tool for the other. But yeah, when it comes to pig diets in the global north, there's not a lot of variation there. You know, there's pretty much a TMR sort of setup, and much more variation would occur in global south countries. So yeah, like I say, I think you're right. We would see different relationships coming through because, well, yeah, monogastric versus ruminant. But yet, once we've got the data, we can look at that more closely. I'm hoping it's a once and not an if we get the data. I'm saying once we get the data. Hopefully.
Thanks, Tony.
Yeah, I would say thank you very much, Tony. I think we have 1.5 hours, unfortunately, for this most important topic. So now we'll go to the next presentation, which is Sasha Hafner from Aarhus University, who will give a presentation with a title, Laboratory Measurements of B0 or B0, Assessments of Reproducibility and Recommendations for Accurate Results, which is really a nice follow-up on the presentation just given by Tony. And very shortly, Sasha, at the time being, or you are working at Aarhus University as an associate professor. You have a long record of carrying out very process-based research, I would like to say, of biogas production, and you are developing models of ammonia emissions and greenhouse gas emissions from manure management for the last 5 to 10 years. And right now, I know you are also very much working hard on how to measure B0 precisely. So, Sasha, the word is yours.
All right, thanks for that, Sven. I guess you all can hear me and see my title slide.
Yes.
I mean, no, not great. So yes, I'm going to talk about laboratory measurement of B0 or B0, or I'm going to call it biochemical methane potential, essentially the same thing. All right, I'll start with a summary. So these are the points that I hope to make. First of all, what we're talking about when we talk about BMP, biochemical methane potential, is the maximum quantity of methane that a substrate can produce during anaerobic digestion. And it's typically measured in batch laboratory incubations. One problem with BMP measurement that I'll focus on is reproducibility. How consistent is a BMP result among laboratories? And Tony, sorry I joined a bit late, but yeah, of course we have discussed your project, and I agree that this is an important part of your project. So if we want to know what BMP is, whether for a research project like that or for a particular substrate to estimate, to predict methane emission, or to determine how much methane we can get during anaerobic digestion, we need to have an idea of how accurate our result is, and ideally we want an accurate result. So there are a lot of different types of methods that can be used, and it seems all of the common methods can produce accurate results. And then I'll end with some recommendations for measuring BMP, including consulting available guidance, selecting an appropriate method, planning tests carefully, and here's an important one, is including a positive control, checking for leaks, applying validation criteria, and of course, participating in interlaboratory tests. So let's get to this material. So BMP or B0 or B0, again, it's the maximum potential quantity of methane that can be recovered from a substrate. Now that perspective is one of trying to produce methane. If you want to recover methane, if you're interested in greenhouse gas emission, it's not such a positive thing, but it's still the maximum amount of methane that could be produced from a substrate through anaerobic digestion. Anaerobic degradation. So it's a pretty simple concept, but in practice, results are variable. So what is the concept? How does it work in laboratory incubations? Well, you take some substrate, say it's cattle manure, and you mix it with inoculum, an active microbial community that includes all the different groups you need to degrade organic matter to methane and carbon dioxide. And then you put it in a sealed container or a system that can measure the quantity of gas produced, and you incubate it over time, maybe 30 days or so. And then you need some method to measure the quantity of methane that's produced in that from this combination of substrate and inoculant. And we have some very high resolution measurements here, quite a smooth curve. So of course, the cumulative total increases over time. Once we have these numbers, we need to subtract the quantity of methane that we think came from the inoculum itself. Because an inoculum, even in the absence of substrate, is still going to produce some methane, both due to degradation of the microbial cells present, but residual substrate as well. And that difference gives us what we might call an SMP curve, specific methane production. For example, in milliliters of methane per gram substrate. And our final value, typically our final value is taken as BMP. You may argue that we should extrapolate to infinite time and call that BMP or B0. We'll get similar results doing that, assuming our SMP curve is typical. So BMP can be measured with different methods. It is measured with quite a diverse set of methods. And as I mentioned, there's this problem with reproducibility. Probably, well, in general, related to a couple points, I think. One is that measuring gas production can be challenging. So this is something that's invisible and does not have a very large mass. It's different from measuring VS, for example. So in general, measuring gas production can be challenging. Another issue is this is a biological assay, right? We depend on the activity of an anaerobic microbial community to measure BMP, and that adds some complexity to BMP measurement. So I've been lucky to be involved in a couple relatively recent interlaboratory studies. These are useful for determining how much variability there is in BMP measurement among laboratories, and that's what I'm calling reproducibility. So what's been done in these studies? A few substrates have been selected. So typically, dry, stable materials. This was some kind of animal feed for rabbits, actually. Cellulose is a pure substance used as a positive control. We know how much methane we should get from that. Sunflower seeds, also dry, quite a different composition than at least the animal feed we used, and then wheat straw. Less degradable and more slowly degradable to challenge inocula a bit. Anyway, the main point of an interlaboratory study is that we have some consistent substrate or substrates that's shared among laboratories. Each of them then applies their method, perhaps with some standardization, to measure BMP, biochemical methane potential. They report results. In our studies, we asked not just for their final BMP value. This is milliliters of methane, standardized to a particular pressure, temperature, of course, dry. This is milliliters of methane per gram of VS, volatile solids, in the substrate. So we asked them to give us their reported, to give us the value that they calculated from their measurements, but then also to provide raw data, which gives us an opportunity to check their calculations. Anyhow, each lab has a value, and we can see just from plotting data that some of the differences are a little too large, more than a little too large to be comfortable with. So here we have more than a twofold difference between the minimum and the maximum. We can look at the overall variability among results by calculating a standard deviation. And then it's typical to express that as a relative value relative to the mean value. And that's one way to quantify reproducibility. So these results from the most recent interlaboratory study show some variability, and reproducibility in this was around 20 to 30% Here's an earlier study for another 4 or so substrates. And this is summarized differently. Instead of a plot, we have, let's see if my pen's working here. Yeah. Instead of a plot, we have a relative standard deviation, relative reproducibility standard deviation. We see 8 to 24%. These numbers perhaps are not terrible, but when we get up in the 20s and 30% for a standard deviation, we're going to see a lot of variability in the BMP value we get. And that, again, reduces the quality of BMP measurement. If we can't be sure that our BMP result is within 10 or 15% of the true value, then it really reduces the quality of our measurements. Another way to look at these data is relative range. So that's shown here, the difference between the minimum and the maximum. And once we get to say 50%, then we're looking at a twofold range in between our minimum and maximum. So there's quite some variability in BMP measurements seen in these interlaboratory studies. Okay, so what's the problem? And the short answer is it's not entirely clear exactly what causes the problem. We do see that diverse methods can give similar results. I've picked a table from one particular study here with results from 2 very different methods. Here are some measurements from a gas density method. I'll talk a bit more about later. This is a method that I recommended to Tony. Very manual, simple method that can give similar results to a sophisticated automated instrument. So within 10% or so for these 3 substrates, a positive control, a mixed substrate, and then wheat straw. So it's not a simple case of saying some methods are no good and others are fine. The differences between methods that cause problems are more subtle. Here's an example where we have some measurements from a gravimetric method, another method that I'm quite fond of. Those are in the gray bars, and then some measurements from a manometric method where the pressure in a bottle headspace is measured over time. And of course, we know that pressure is related to, even directly proportional to, the quantity of gas that we've forced into a bottle, or that the microbial community has forced into a bottle. We've found that results don't always fit theory, though. Here on the x-axis, we have headspace as a volume fraction. And as our headspace volume gets smaller, that's moving to the left here, we see this bias in manometric measurements. We see a lower value for the manometric measurements compared to gravimetric and to the overall mean. So let me jump right to recommendations for measuring BMP, and I'll spend the rest of my time in this presentation talking about recommendations. So I've read this list before, I won't read it again, but I'll start with the first point. So for available guidance, a group of biogas researchers involved in this first interlaboratory study that I mentioned, and of course there are others that exist apart from the 2 that I mentioned, but a group of researchers compiled a bunch of free guidance on a website we've called the Standard BMP Methods website, perhaps with a hopeful perspective and maybe not a modest enough perspective, but this is the term we chose. So it's loaded with free guidance on BMP measurement. You can enter this URL, or if you have a phone open, you can scan this QR code right now and check that out. This is what it looks like. And we've got info, basic information to all kinds of advanced information. And I want to highlight this method section, which has documents on both laboratory protocols and calculations. The literature section has quite a few papers listed and links, especially if they're open access, for the papers that this group that we thought were most important. So if you're interested in measuring BMP, check out this site. There's a lot of guidance available now. All right, but how about the method? What method should you use? So if you were to ask for my opinion, you all haven't asked, but I've been given an opportunity to speak here, so I'll tell you my opinion and remind you that others may have different opinions. I'll give you 3 recommendations. If you have funding, then go for a good automated system. BPC is a company in Sweden that produces this AMPTS system. Now in the 3rd generation, this is what the 3 is for. And this gives reliable, accurate measurements. And importantly, it's automated. It doesn't require a lot of effort to run once it's set up. So if you can afford it, I'd recommend that. If you don't, now we're at B, but you do have a gas chromatograph or some other reliable method for measuring biogas composition, I prefer gas chromatographs, then use the gravimetric method. In fact, I would say this is probably, in my opinion, this is probably the most accurate BMP method. This is where you measure the mass loss of bottles over time. Biogas does not weigh much, but it does, of course, have a density, and typical laboratory scales are sensitive enough to quantify it. And it's difficult to trick a scale. You can easily trick a syringe or an inverted graduated cylinder by having a gas leak or by not measuring at a consistent pressure. So you can trick those methods, but it's difficult to miss mass loss when you're using a scale and you're checking for consistency over time. So that's a great manual method. It requires manual measurements over time. If you don't have access to gas chromatography, now we're at point C, then I'd recommend the gas density method. So this is a very simple method that can still give accurate results. The idea here is that you measure both mass loss. So here we've got a bottle set up with the substrate and inoculum. It's incubated. It's taken out of the incubator intermittently. At first, this would typically be daily over the first few days. And it's weighed. Then biogas is removed using a syringe, and that volume is recorded, and then it's weighed again. Again, biogas does not weigh a lot. What are we around? Somewhere around one gram per liter. It does have a mass, and you can weigh that mass loss. So in this case, by comparing this and this mass, you can determine the mass loss. You have the volume here. From that, you get the gas density, and biogas density depends strongly on the composition because carbon dioxide weighs much more on a molar basis than methane. So from that, you can determine the composition. Seems like a crazy idea, but it actually works all right. So what else would I recommend? You should leak check manual methods with a scale. So even if you don't use a gravimetric method or the gas density method that of course automatically includes weighing, if you go with volumetric or manometric methods, pressure or direct volume measurement, then you can always weigh your bottles at the start and at the end, and you can compare mass loss to what you would expect for mass loss based on your biogas quantity measurement. These are some results from an interlaboratory study that showed quite a few measurements were below the 1-to-1 line that we would expect if there was no leakage. In fact, I think leakage is quite common, not always large. This dashed line is perhaps 20%, but there are some observations around 20%. It's easy to check with a scale, so do it because it does happen. For BMP test planning, one of my recommendations is to carefully plan BMP tests. And in particular, you could argue that perhaps this isn't about planning. Anyway, I've listed it here. You've got to select a good inoculum. And I think this is often a limitation and a contributor to poor BMP results. Ideally, this inoculum should come from a full-scale anaerobic reactor that is that's operating well. If that's not possible, you can develop an inoculum in your laboratory. And I'm happy to give advice by email or anyway on that suggestion. You need an appropriate quantity of substrate. 1 gram of substrate VS at least is a common rule of thumb. You need enough inoculum, and that's quantified by the inoculum to substrate ratio on a VS basis. And that's typically, if you're unsure what to use, typically 2 to 1. So twice as much VS comes from the inoculum. Your headspace needs to be big enough, largely for practical reasons, to avoid too much pressure or foaming issues or exploding bottles. And it's important, it's essential, I'd say, to include a positive control. Some substrate with a known BMP to check all the components of your method. So for balancing, for let's say, for considering multiple constraints when planning a BMP test, you can use this web app called OBA for the online biogas app that does have a BMP planning component. This is a free web app that lets you enter your constraints and then gives you test design, just the quantity of the individual ingredients that meets all those constraints. All right, looks like I've been talking for almost 20 minutes and
Yes.
I should try to wrap this up. So let's talk about positive controls a little bit. Microcrystalline cellulose is the most common, but there are others that could be used. Validation criteria. These are quite important. So you can find some criteria in the links given in the Standard BMP Methods website. The idea is to check your results after you've carried out the test, and this is where the positive control comes in. So you check the BMP values of your positive control, and if they're within this range, 340 to 395, there are some alternative proposals, and you can say your test is validated. There are other criteria as well. If not, it's rejected. Check out the guidelines again, repeat, and try to figure out and address the problem. So validation criteria are quite helpful for flagging bad tests. Another way you can look at your results is to check the shape of the SMP, specific methane potential curve. So here are some results from that most recent study. And sometimes strange curves, so this one went like this, it's negative, it doesn't fit the typical shape that you see in the lines with color here. Strange curves can indicate problems. And the last recommendation was to participate in interlaboratory tests if you can. If you can't find a test that someone else is organizing, you can organize one yourself. You just need a substrate that's stable to share. Thanks for your attention. I'm happy to answer questions.
Thank you very much, Sasha. Any questions?
We do have questions. Oh.
I could start very briefly. Sasha, what about characterizing the VS? Because we know some VS, there's some organic matters has a lot of volatile products that can be lost during your measurements, and that's not included. Do you have any comments to that?
Yeah, so it is a good point. First of all, the name VS in this issue with volatile components is confusing, I think. But you're right. There are some components. The name VS, the volatile in VS refers to 550 degrees Celsius. Organic matter is volatile there, but there are components that are volatile during the 100 or 105 degree drying step that are then not counted as VS. They come out as water. So alcohols, volatile fatty acids. That's true. That introduces VS measurement error and can give you even impossibly high BMP values. I don't think that's such an issue for reproducibility. It is for accuracy. One way around that is to express BMP on a fresh mass basis or as received, right? And ultimately, when it comes to biogas plant operations, that can be a useful way to express the value. And I'm actually glad you brought that up, because most interlaboratory studies are unable to address that because we use nice, stable, dry substrates that don't have alcohol, for example, and not even VFAs. So it's true, that's a source of error as well. But measurement of VS in general, we found, of course, that's a part of these interlaboratory tests because it's needed for BMP measurement. We found that that's quite reproducible. So that multi-step process that does not have a biological component is quite reproducible, fortunately.
Thank you.
Sasha, there's a question from John. He would like to know how temperature effect is considered in the setup.
Yeah, another good question. So it's difficult to assess. I don't think there is, from the little data available, the laboratories that used different temperatures, I don't think there's much evidence that temperature has a big effect on BMP. That is the final, the ultimate methane potential, as long as it's a typical anaerobic digester temperature, right? So in most of these laboratories carried out a test somewhere between 35 and 40 degrees Celsius. And even if a thermophilic temperature were used of 55 degrees or so, and I actually can't remember if anyone used that temperature in either of these tests. I do think so. Even at a thermophilic temperature, it seems you get faster biogas production, perhaps, but not a greater quantity. Henrik, who will speak next, he's pointed out to me that at much lower temperatures, there are some components that are degraded more slowly, particularly lipids. But as long as you're at 35 degrees, degradation should be and seems to be more or less complete. So it doesn't seem to have a big effect, but we don't have a lot of data in these tests at different temperatures to truly evaluate that question. I don't think it's an issue, though.
There's also one question. I'm not sure if I'll be pronouncing the name right. They're asking what you say about duration of BMP tests.
Yeah, good question. Duration. It's a point I missed. So the community recommendation is to determine, to run a test until the rate of methane production drops below a certain level. And the recommendation is that is for 1% of the net cumulative, or the cumulative net methane production per day. So once daily production is under 1% of the total amount that's been produced up to that point, then you can stop the test. And that typically gives, that typically requires, well, say 2 weeks, maybe 20 days, sometimes longer. But that is a duration that considers or responds to the degradation rate or the activity of the inoculum. So it seems to work better than a fixed duration.
Well, I would have, if there are no other questions, I would have one. It is the questions to the microorganisms which should be adapted. So if you, for example, send out the inoculum to the people who are carrying out the test, they may have a substrate, let's say manure, that is very different from the one to which the microorganisms are adapted, I would think. And then maybe you get results that are not really valid in compared to if you had adapted microorganisms.
Yep, it could be. We don't see a lot of evidence of that. So
Okay.
one of these studies included inoculum exchange, and that's difficult. It's difficult to ship out inoculum because it has a high moisture content, it has a microbial community, and you have issues with customs getting it from country to country. But anyhow, inoculum doesn't seem to explain much of the variability in BMP, as long as it's a decent inoculum, right? Some labs have trouble getting any good inoculum, but in general, as long as the lab has an inoculum from an active reactor, it seems to work well. It does make sense that it could happen, but remember here, we're not so concerned about rates, right? So maybe an inoculum is going to be a little slower than another inoculum because it hasn't degraded much of particular substrate, I don't know, lipids before. But as long as it's from a reactor that sees some kind of a mix, and as long as there's not a toxicity issue, for example, you're digesting a manure that has a very high ammonia content, then it doesn't seem to be a big problem.
Okay, thank you.
All right, well, I'll stop sharing.
Yes, thank you very much, Satya.
You're welcome.
So we are going to the last and presentation by Henrik Møller, giving a speech about overview of a large data set for chemical composition and B0 of cattle manure with different feedings and feed additives. Henrik Møller is from Aarhus University, and he has a background, I think, which is really good for this kind of studies, from being actually a practitioner, setting up biogas plants and running them. And then later he has been working with and developing biogas digesters and also the use of biomass for the last 20-25 years as a researcher at the University of Aarhus, and he is now a professor there. So I give the word to you, Henrik.
Can you hear me?
You can begin, please.
Yeah. Can you all hear me?
No.
So you cannot hear me.
You are not very clear.
Just a minute.
No.
Can you hear me now?
Yes, good. Thank you.
Okay, very good. And is my screen sharing? That's not okay yet, I guess.
Yes.
So can you see my screen?
Yes.
And it's in presentation mode.
Yes, it is.
Yes.
Henrik,
No.
it's on the presenters.
Oh, yeah, sorry.
But how do you change this? Yeah, that looks right. If you switch that, there we go.
Sorry.
Is that better? Okay.
Yes.
And you can see my mouse also?
Yes.
Yes.
Yes. Okay. So thank you for the possibility to tell a little about our experiments, especially with cattle manure. And as Sven told me, I come from the University at Aarhus. And the good thing here, when we are doing all these BMP tests and so on cattle manure, is that we have our own biogas facility running at a high share of cattle manure. So the inoculum issue is perfect. Then we have a farm with around 400 cows where we are doing a lot of feeding experiments, and we are doing a lot of experiment with enteric fermentation and so on. So we can relate all the things together. Just a short introduction to the biomass in the Danish agro biogas. As you can see on a mass level, cattle manure is by far the most important biomass. So that's why we have a special interest in the cattle manure. And also there's a lot of incentives in the Danish agro industry to reduce enteric fermentation and the combination with biogas. So when we talk about cattle manure, the feeding is very different. And of course we have two routes in Denmark around, I think, for the cattle manure. In general, around 40% of the manure already is going to the biogas industry today. For the cattle manure, I would say it probably maybe close to 50%. So a very high share of cattle manure goes to biogas today. And there's a lot of interest in Denmark to reduce the enteric fermentation and see how the downstream processes is affected by changing the feeding system and so on. In general, when we talk BMP, we can sample different places. But the problem is that we have losses all around, all the way through the chain. We have losses in the building, which is different from every farm. We have losses at the pre-storage before the biogas and so on. So what we do when we characterize cattle manure is that we very often look at the animal level. We take what we call a grab sample. That means that you put on a glove and take your hand in the back of the cow and take out a portion of the feces. And in that way, it's the feces that we are looking at. And this means that we can also do it very homogeneous because it's not mixed with urine and we don't have to mix it afterwards and so on. So it's a very good way. Beside the feeding, we are also looking very much on additives, how that affect the BMP or the B0, because that's important for the downstream processes. Already from this year, as the first country in the world, it's been mandatory to add enteric fermentation inhibitors, mainly the Bovaer, the 3-NOP, which has to be added to all cows this year. Unless you use the alternative, which hardly any farmers do, that is to add a lot of fat to the cows, which could be an alternative, but more expensive and until now not selected by very many farmers. So the BMP, let's see, where's my mouse here? Yeah, we do it with the water displacement methods. We have done that for 25 years now, and it's very well established in our lab. We analyze the gas composition on a gas chromatograph. We always freeze the samples. That's necessary because we want to have a lot of samples before starting up a BMP when we have to compare, because there will be a experimental factor in every BMP. If we take the same sample in two BMPs, there will be a small variation. So because of that, we want to run like 30, 40 samples at a time to compare them, the different feeding strategies in the same experiment. And as I say, we mainly do it without the urine because we make these grab samples from the cows. We almost always use thermophilic inoculum. We have tested it, and I must say that especially for cattle manure, there is a temperature impact. We see higher yields also after. We always do it 90 days. We will always be in all our experiments. We are below what Sasha mentioned as a daily increase. When we do 90 days, it's a long time, but still we are sure that we are when there's hardly any increase anymore. For the B0 value, we most often use the first order kinetic model. We always do it in triplicates, and we calculate the B0 value. And we also calculate the hydrolysis rates. I think for emission issues, that could be an important factor, which we don't know yet how much means, but it could be an important factor. In some cases, when we have a lag factor lambda, we also use the modified Gompers equation, so to calculate the B0 if we have a lag factor. Often we do both models also and calculate, because then we have the lag factor included as a parameter also. So during the years, we have done a lot of experiment with cattle manure and BMPs. So this is an example of the amount of samples we have measured during the last years, during the last many years. Actually, we are doing a lot at the moment. We have a new project in Denmark where we are sampling a lot of samples, which is not, I don't have a result yet, but they will come in the close future. So looking at all these samples, we always measure most of the chemical components like the cellulose, the hemicellulose, the lignin, the volatile fatty acid, the protein, and lipid and residuals. And as you can see, there is a rather high variation in the different components. For the cellulose, it's rather constant. Same for hemicellulose, with some variation. But when we go to lignin, there's a high variation. And for the lipid and residuals, there's also very high variation. We have made an average of all these components. The first figure here is in terms of volatile solid, but we also have the ash, which is around 13% of the dry matter is ash. And including that, we will see some average composition shown here from a large array of cattle manures. And of course, with a huge variation in some of the factors. Yeah. So just an example of the impact of diets on the BMP. This is an example that's published quite some years ago when we looked at different diets, like in this case, a lot of diets with addition of fat also, early grass plus fat, late grass plus fat, maize diets without additional fat, and maize with fat. And then some different and experiment 2, which is mainly the different kind of fat additions to the cows. And we made some correlation analysis in the 2 experiments, experiment 1 and experiment 2 here, and then we pool the 2 experiments to see if we will have a correlation. And we have tried to correlate it with the diet composition and the feces composition. And the factors where we found some significance in the diet, that was the fatty acids and the gross energy and the crude fats. In the feces composition, it was only the lipid and the lignin, and in a few examples, the volatile fatty acids that gave some significance. We looked at both the biogas and the BMP in the 2 experiments, and time is running, so I'll mainly focus on the pooled experiment here from the 2 experiments. And what we can see here, the B0 value, there's a quite good correlation with the fatty acids in the feeds, the gross energy, and the crude fat in the diet. Looking at the feces composition, it's mainly the lipid that plays a role when we look at BMP. It was quite surprising that when we pooled the 2 experiments, the lignin was not significant. The lignin was significant in some cases in experiment 2, but not in experiment 1. Then we tried to make some models with the different components. We made a diet model, so only knowing what has the cow been fed. And we have all these components, a constant and a gross energy, crude fat, fatty acids, NDF, and crude protein. That was the one who made some significance in the diet model. Then we had a pure feces model, and then we made a combined model of the 2, where we both use the diet and the feces components. And as you can see here, we could have some quite good model, including the different components. The best model is combining the diet and the feces. And here, if we have these components like the gross energy, fatty acids, NDF, and crude protein, combined with the VFA and protein diphases, quite surprising, only these two, we had an extremely good correlation for predicting the BMP. But I think it would be nice to include this to much more samples. And we will do that in the future to try to get some better models. One of the things was that we were very interested to relate the BMP to fat, because fat has an extremely good effect on reducing enteric fermentation. So if you add sufficient amount of fat, it's as good as the chemical components like the Bovaer. So, but what we saw also is that adding the fat, we have a quite good correlation with the BMP. You can see here we have both at day 30, day 60, and the day 90. I think, yeah. And you can also see that a lot of people only do maybe to 30 or 60 days. You see a lot happen from 30 to 60 days. So for cattle manure, I would say you should go for at least 60 days. Even going from 60 to 90 days, you still have around 10% additional methane. It's very time consuming, but if it's a real BMP you want, I think you need for a very, very long time for cattle manure. And this is under thermophilic condition. For mesophilic, it will be quite much longer. And we also see that in general, we do see lower B0 values for mesophilic, even with very long retention time. So we also correlated the fecal methane potential in liter methane per kilo dry methane intake compared to the enteric methane production. So if we increase the amount of fat, we have a very good impact on the enteric. At the same time, we'll have a very good impact on the methane yield, the B0. So that is actually, in a way, positive, as long as you make biogas of it. If you just store it, the high B0 value would be a drawback. Also, we have a good correlation between fat and both in feces and in diet on the B0 value. So for the farmers that deliver to a biogas plant, as long as the biogas plant will put value on it, that these increasing amount of fat increase the methane potential for biogas and have this huge impact on enteric. We also looked at the lignin, and it was a little. I need to go a little more into modeling some of this, but the pure lignin, that's which we actually expected would be a quite good indicator. Yeah, there's hardly any real significance on the BMP. So of course, the very, very low lignin has a high yield, but when we come beyond a certain level, like more than 10% lignin, then it's a very, very weak correlation between lignin, which is actually quite surprising. So for all these experiments we have done, I just make the average also here, and for Danish dairy cattle, for sure we are on a higher level than the IPCC values. So for very high productive system like Denmark, I think that the IPCC is underestimated. Then a little about the impact of additives. Here we have a study made some years ago where we compared low fat, high fat levels and combined it with the additives. There's two additives that is of main interest in Denmark. That's the Bovaer that I already mentioned. And then we have the silver, which is nitrate, which also have a quite good effect on enteric fermentation. The conclusion is at a low fat level here, for example, that there's really no significance on the treatment. That means that adding both Bovaer and Silvair, it doesn't give either higher or lower methane potential or B0 values. On the other hand, if we go to a high fat level, we have a high jump in the level of BMP or B0 with more in general around 18% higher gas yield. But at the same time, there was no impact on the combination on fat with the additives either up or down. We are doing a lot of more experiment with that at the moment. But I don't have the result yet. So conclusion and summing up. So cattle manure is extremely important for the Danish biogas industry, and therefore we have a high interest in it. We have this ultimate gas yield at around 285 liter, and an indication that the gas yield is very variable and impacted by both diet and feces composition. It seems that we can get some rather good model of combining models of diet and feces composition to predict ultimate gas yield. And then finally, the chemical additives, this has no significant impact on the ultimate gas yield. So thank you for attending, or for listening.
Thank you very much, Henrik. Any questions for this interesting presentation?
There's a question from Andrej. Could degradability of fiber fractions NDF, ADF in the rumen have the effect on the BMP?
Could you please repeat it? I didn't got the first part.
Could the degradability of fiber fractions, the NDF, ADF in the rumen have the effect on the BMP?
Yes, it seems so. It seems so. We don't have the exact correlation, but it does seem so that, for example, early grass, contra late grass, and so on, so have an impact. So fiber composition have an impact. We don't have a very good correlation at this moment from the experiment we have done, but until now.
Tony has a question.
I'll just jump in since I've got access to the microphone. Bit lucky. That's great. Thank you, Henrik, for that presentation. It's very informative and great to see that you're, for me, sitting on a very healthy looking data set, I have to say. And I wondered if you've done a lot of work here with cattle. Have you done work with swine as well to the same extent in terms of impact of diet on BMP?
Yes, not to the same extent at all, but we have recently done it for some kind extreme diets with a control diet, and then with quite extremes. So we have data. It has just been published recently by Elvira Satori from our institute. So that gives an indication. Then we have a lot of non-published data from the industry, but the real challenge there is the sampling, I would say, because here we usually don't take these samples directly from the feces. We often take it from the slurry channels, and there we already have some losses, and we have a dramatically increase in VFA very fast. So that's we don't have so good value because of these issues, and also the sampling. That is an extremely important issue also for your study, Tony, or your project. So the sampling, if you are sampling in a slurry pit, it is challenging.
Okay, I've got a supplementary question if I can be so cheeky. The feed quality, I would assume is very good, very high for your Danish livestock systems. Do you have much? How broad is that data set in terms of feed quality going to more sort of roughage type diets? Does it capture those sort of dietary systems as well?
Yes, they do.
Okay.
It's quite some of the data are quite some years ago, and it's something I would try to dig a little more into. But we have some the roughest quality, especially of grass. We have some study on that.
Okay, good to know.
So
Thank you. I
but
won't take any more
it is high yielding. Of course, it's very high yielding system, all of it. But with
Great, thanks.
and we are doing a little bit high heifers also now. I could see April's results that they are very, very different from heifers to dairy cows. So that would
Yes,
be
she put that down to the diet. In terms of not so much the age class of the animal, it was the diet that they were on giving that result. Yeah. Thanks.
Henrik, there's 2 questions, actually 3 now. One from Daniel. I sent you on the chat. I think it'll be easier. Can you see it?
If I can see the question, I'm not.
Yeah, on the chat.
Let's see. From John, from Daniel Muller. Yeah, hi, this is Daniel Muller. Yeah, thanks for the. Do you have any on the use of animal feed rich with condensed tannin on B0? No. So I'm not very sure about this tannin. Is that an additive, or I guess it is, but sorry, I don't know much about tannin, and I don't think we have any data on that now. And then another question from Melissa: what is the optimum number of days and what factors influence also when does peak typically occur? Yeah, of course, that's also a little too search, but for we have chosen that in general we always do it for 90 days, even though it's in some cases it's too long. I think it's easier for us just to have a fixed day, and the highest the period with the highest yield per day. It differs a little, but I would say maybe after 4 or 5 days. That is so sometime in the start when everything is adapted, and yeah. But I can't answer exactly on it, and it differs between experiment when you have the peak rates of methane production. Yeah.
If there are no other questions, I have one. You were saying, Henrik, that the production, the BMP or B0 measured differed between mesophilic and thermophilic, and now these B0s or BMPs they are using to calculate emissions from liquid manure storages. So could that actually affect, which methods to measure BMP would? Do you think that would greatly affect the calculations of emissions nationwide?
It could be. It seems that for the very lignocellulosic biomasses, From my point of view, temperature seems to, we have some hydrolytic bacteria that seems to work at thermophilic temperature, which doesn't really work good at mesophilic. So we have a shift at the thermophilic. So even for 90 days, we see maybe, I don't have a lot of data on it, but we see maybe 3-5% extra gas for the thermophilic. And even more in some cases with high amount of lignocellulose. So temperature do have an impact, and how it's then related to the final emission in the storage without biogas. Yeah, that's another question. What is best to use? Should we use the mesophilic or should we use the thermophilic, and what is it we want the B0 to be used for? Is it really the maximum bacterial capacity? Then I think we need to go to thermophilic because there is some hydrolysis processes going on there, which not, especially also on lignocellulose and also in some cases of fat.
Thank you very much.
Yep.
So if there are no more questions, I'll wait a little bit.
Let's stop sharing.
Yeah. Then there are no more questions. I think I will really say thank you very much to Henrik, to Tony and Sasha, and to the GRS Secretariat. I forget names, I'm an old man. But this has been a most interesting webinar, and I'm so glad that you would participate and contribute. Tony, Sasha, getting up in the middle of the night, and all of you giving so nice presentation. I'll just mention that we'll have a new manual management webinar the 16th of June about actually greenhouse gas emissions in sub-Sahara Africa, which follows up quite much on the work that Tony is doing. I think the idea is that the 3 presenters will show what is known and what should be known, and maybe they would also invite for collaboration. So maybe if you might be interested, then the 16th of June there will be one more manual management network webinar. And if you yourself, the audience, is really strange not to be able to see the audience, but I really, I hope that some of you will be interested in giving one of these webinars. And then I and the Secretariat will do a lot to help you so that it's not too much work for you to go together and give 2 or 3 or 4 presentations, and that can start some nice collaboration, I think, or deliver some knowledge that is needed. So you're so welcome to contact me or to contact the Secretariat about ideas, and we'll do everything to help you to make it not too laborious for you to organize these webinars. So thank you very much for listening. And goodbye and good night, as they are saying.
Thank you, Sven.
Yeah, you're welcome.
Thanks, Sven.
Thank you, everyone. Bye.
Goodbye.